Good morning, everyone, and welcome to the first day of our MPG 24-25 season. My name is Nicole Rockweiler. I'm a postdoc in Steve McCarroll's lab. Myself and Michelle, we co-organize the MPG Primer, and we're so glad that you guys are tuning in today remotely or in person. Just wanted to give a few slides for housekeeping information. So as you I've already can tell we're in a different location this year, and this is just to make it easier so we can transition to the seminar meeting. So we'll be here in this room every week unless there's a room reservation issue, but you can always check our calendar. And then also just to be cognizant that there's another seminar right after this. We're going to end at 9:20. Feel free to stay here if you want, but if you want to move over to get your next breakfast and get a good seat for the seminar, just feel free to do that. And also just want to tell you today and forever, we really encourage you guys to ask your questions. This primer is for you. If something is unclear, you want to know more, please ask your questions either at the mics or put it in the Q&A on Zoom. And then lastly, we put these meetings online afterwards on YouTube, so they're open meetings. And then again, this year, we're going to start something new. We're going to try to put these PDFs of the slides on the Broad website that's publicly available. So if you want to share with colleagues, it's another resource for you. One more bookkeeping thing about today. Today we have our primer, our seminar at 9:30, and then the next event today is a happy hour with food and drinks. It will be at 4 to 5:30 in the terrace, just across the side. And if I didn't mention, both food and alcoholic and non-alcoholic beverages. And then another update, we're trying a lot of new things this year. We are officially on Twitter as Broad MPG. So if you want to learn about our primers when the videos go up, read about papers from our community as well as job postings, please consider following us and also tag us with your research so that we can follow you and share your work. And you might notice one less person in the room right now. Lauren, who was our program manager, she actually stepped down from the position. So we are actively looking for a new person to be the operations specialist for MPG and TGG. The job is officially posted. There's this IO link here. If you have any questions about the position or know someone, please let myself, Heidi, Mark, Michelle, know. And also in the interim, while Lauren's out, there's this email address that you can send if you have any questions about MPG in general. I'm going to skip the description. I'm going to post it on Slack later today. But please, if you know anyone, we would love to have them on board. So just to give you a preview of what's coming up in the beginning of this primer season, we're going to really focus on basics of population genetics. So next week, we're going to have an intro to PopGen, and the following week, we're going to do more PopGen topics such as population structure, stratification, and admixture. And lastly, I'm pretty sure this is the last slide. Today, we have a lovely introduction to R for data analysis by Nil, and Michelle's going to introduce her.
Great. Thanks. Good morning. It is my pleasure to introduce Nil Aygun. Nil received her PhD in bioinformatics and computational biology at UNC Chapel Hill in 2023. During this training, under the mentorship of Dr. Jason Stein, she investigated genetically altered cell type specific gene regulation during human neural differentiation. She began her postdoc research in the Barocum lab in May 2023. And in her postdoc, she's developing algorithms to explore the genetic and other molecular co-occurrences driving cancer progression and drug resistance. But today she's going to be talking to us about data science with R, an introduction. So with that, I hand it over to Nil.
Great. Thank you so much. Yeah, today I would like to share with you some introduction for how to use R in data science in general. So I'm really happy to be here today. Yeah, today we will go over some basics in R and how to use them in specific genomic analysis. And this is our outline. We will learn basic data structures, how to manipulate data in R, how to define specific functions, how to use control structures, how to perform regression analysis, and how to visualize your data. And some there's specific specialized R packages you can also use for genomic analysis in addition to what we go over today. So to be able to give you an introduction all of these topics, I would like to start with an interesting genomic question so that we can introduce certain data set and how we can answer a specific questions by using R. So this question includes some QTL work from my PhD study. So in that, as you're all familiar with, genome-wide association studies discover many inherited genetic variants that are associated with a variety of complex human traits. One interesting fact is that those variants are mainly found in non-coding part of the genome, and that makes hard to explain their function. So one of the potential mechanisms is that a genetic variant can be located promoter of a gene that may interfere with binding of transcription regulators, and that will change the chromatin accessibility in that area. And accordingly, based on the having different variants, different allele of this variant, we can see changes in expression as an outcome. So by performing expression quantitative locus ensured QTL analysis, we can try to understand if the individuals that have AA genotype has a higher gene expression than individuals with CC genotype. And in this case, we perform a statistical test and understand if this relationship is significant. So let's say we are interested in performing expression quantitative locus analysis. And these are the, oops, let me go, how can I go further? Oh, okay, here we go. So let's say we drive some data sets from this to perform this analysis such that we perform RNA sequencing by collecting hundreds of individuals and drive cell lines from them because we are interested in cell type specificity. And we perform genomic transcriptional profiling across the cell lines by performing RNA sequencing analysis. And here is the data set we can see for gene expression. In this case, each row indicates different genes and their corresponding expression value across those cell lines coming from different individuals. And you can see that those values correspond to normalized gene expression value. And we can also look genotypes of the cell lines derived from these individuals. In this case, you can see the variety of genetic variants listed on the row and their corresponding genotype information across cell lines. For instance, one indicates that in my case, I call effect allele as a minor allele. And if an individual has only one minor allele, we can call this a 1. If it doesn't have any, we can call this a 0. If it has 2 of the minor allele, we can call 2. So this is a numeric interpretation of how a genotyping data set can be done for a QTL and any type of variant association analysis. In addition to that, we may have a metadata which includes information from the cell lines, which can be further useful for us. For example, let's say we sorted cells based on a certain marker by using fax machine. And unfortunately, there are 2 different fax machines were available. That's why it caused some batch effects. That's what we can call them fax machine A and B. And those individuals, they provided cell lines for us, may have different genders, male, female. And you can also store their ages as well. So, yeah, to be able to import this data set and work on them in R, we first need to understand data structures, potential data structures in R. Let's look at them a little quick. Vectors are the basics units how to represent data structure in R. It basically includes a list of elements, and by using C parentheses, you can define a new vector. In this case, I define a vector named A, which includes 8 numeric components. And so this is how you assign a variable in R by using equal dash operators. And this is how it looks like. And you can also check what type of data is by using a class function in R again. As you see that it was only numeric values. In addition to numeric values, we can store character, or in other words, string components in a vector. In this case, these are different gene names, as you can see, and there's a character vector. And matrices is a 2D form of the vectors. In this case, you can store multiple information from multiple vectors. As an example here, you have 2 different vectors, A and B, that includes numeric values with 3 different components. By using cbind function, in other words, column bind function, you can bind this vector by column that result in this 2D information. And we can check the dimensions of this matrix by using dim function again. Alternatively, you can define a matrix by using a matrix function. In this case, you provide a vector as a first parameter of this function, and you can actually, the type here is 3, and you can say that how many rows you expect in your matrix, and if they are ordered by row or column. So to be able to access elements or slice or subset of matrices, there are certain ways. So for this, we can use square brackets for our objects. In this case, our matrix object called as M. So let's say we want to interested in to access elements which located second row and first column of this matrix. For this, we can define first element before comma as the row information and the second one as the column information. Alternatively, we can get, for example, slice the matrix so that we can get only the first column and then all the information from the rest of rows. We can basically leave empty the first item, and second place can be the number of the column you're interested in. Data frames are a little bit more complicated version of the matrices. Difference between matrix and the data frame is that you can store a variety of not the same data set, but different structures, different classes, excuse me. For example, you can store both numeric and character information in the same data frame. Such that, for example, let's back to our previous example, we have cell lines indicated by their identifiers here, and the text machine which is used to sort them, and gender information and age information. By using the data.frame function, you can define a data frame. As you can see, you can place each of those vectors, and they result in a mix of information coming from numeric values and character values. Again, you can slice or subset data frames by using a similar strategy we use for matrices, by using a square brackets and writing the name of the columns for row interest. For example, let's say I want to get the values from first column to second column by keeping all information from all rows. I can put 1 column and 2, and this will give me the information from the first column, second column. And also we can select information from the second and third rows across all the columns. So this, now we will place our interest range to the first place, and then leave the empty in the second item. And alternatively, how we can call certain columns from data frame can be performed by using a $sign. For example, each column has a name in our data frame, and by using a $sign, we can pull information from fax machine. And that will result in, as you see in a vector, it's a character vector, and str is another function. You can learn what type of data set you have within your vector or in general in your data frame. And lists will be the final data structure I will introduce you here today. This is an even more complicated version of a data frame, which includes a vector, a character, and even a matrix within a same object. So in this case, I included a numeric vector called numbers, and a string, a word has genetics, and a matrix which has 2 columns and 3 rows in the same list. And eventually you can see that all of them can be stored in the same object. To be able to extract the elements of the list, in this case, you need to use double square brackets. For example, if I want to reach the matrix within this list, I can use double square brackets, and also writing the index of this where this matrix is stored in the list, which is 3. And as you can see, again, I can look for this by using str. I can learn what type of a data I have here. That indicates that I have a matrix which includes numeric values. And also similar to a data frame, you can use a $sign to extract an element as long as you have a name for it within your list. And again, you will extract your matrix information within that list by using $sign matrix command. So after learning those data structures, let's look at how to manipulate data in R. And we will go over that by asking some very simple questions. And before going that, I would like to introduce you tidyverse, which is a collection of different R packages that is designed for data science usage and which can be used for data manipulation. As you notice, I said something different here, which is package. So let me first introduce what is package, what does it mean in R. So in addition to certain function which you can find in base by using base R without need for any other help, I would say, you can perform many tasks. But however, there are also packages in R. They can be add on all of this base R functions, and that allows you to perform additional tasks. So you need to install these R packages as you need. And there are 3 major resources you can install these packages. One of them is current. And by using install.packages function, you can install your package of interest. In this case, for example, tidyverse can be installed in that way. Bioconductor is another place which includes our packages that are specified for biological functions. And by using BioManager, you can install these packages as well. So BioManager is another R package which allows you to install packages stored in the bioconductor. So that's why you first need to get the BioManager from current, and then you need to install packages from bioconductor by using the help of BioManager. And GitHub is another place you can also get the data. For that, you can use DevTools, which is another R package, because this is a very dynamic field, and every day people generate their own R packages, and they don't prefer going to apply bioconductor for each time. That's why they just place them in GitHub because it's free, open to the public. So basically, you can, if you are interested in to use their own packages, you can download them from GitHub based on the addresses they provide you. So one important thing here Installing a package is only a one-time task. Once you install a package in your R, you no longer need to do that. However, what you need to do each R session is you need to load them. For this, you can use a library function, and every time once you want to use some functions from a package, you need to you need to load in your environment by using this library function. So let's go back to data manipulation part again. So in this case, we want to answer such a question like which cell lines in our data set were sorted by fax machine A. So for this, first we need to import the data set of interest in our R environment. For that, we can use read CSV function because I have a CSV function. It can be an Excel function. You can use a different Excel document. You can use a different function for that specified. Or you may have a text file. In this case, you can read table, or you can use fread function, which is coming from data table package, which is very helpful once you have a big data set. It requires high memory as an alternative. So to be able to read this data set, first we need to define where is the data set. This is how I store my data set, called as metadata, and then you write the name of the file. And once you read this file, you need to assign another variable so that you can work with as an object in your R environment. So we can look at what is inside in this metadata object by using head function. That will give you the first 6 rows. So you don't need to look at the full data set, for example. Again, you can see the cell line information and the corresponding fax machine, gender, and age. So now if you go back to our question, that is, we want to learn which cell lines were sorted by fax machine A, excuse me. What we can do is we can use this specified functions from tidyverse, more specifically dplyr. So now I would like to introduce you this operator, which looks a bit complicated, which is called as a pipe. So that allows you to use an output from your previous step as an input for your next. In this case, I will use a filter function so that I can get only rows which has a in their fax machine information. So that's why I define it in here, and then that resulted in the subset this metadata so that only information is kept where cell lines were sorted based on fax machine A. As a second step, what I would like to learn is I want to learn which cell lines actually have this information. That's why I only need a column that includes the cell line information. To be able to perform this task, I need an additional comment. Again, I add my pipe here, and I will perform that by using the select function. So within the select function, I'm writing the name of the column I'm interested in to extract, and that will result in cell lines that were sorted based on fax machine A. So let's ask another question. That is, I'm going to learn what is the age average of females versus male donors from which cell lines were generated. For that, again, my data set metadata is here. I want to group them based on their gender so that I can get a gender specific information by using group by function. And then from the separated stratified data based on gender, I want to calculate average of the age. To be able to perform that, I can use a summarize function. And within the summarize function, I will write my function of interest. In this case, that is getting the mean, calculate the mean for the column of interest, that is age, and that will result in average age for females and males. What else I can do? For example, I may also be interested in adding additional information for this metadata, let's say ancestry information. And we have a list of ancestors for each of these donors that we got the cell lines from, which is an object called as ancestry, it's a character vector. And to be able to add this information to my data set, I can use mutate function, again, after the pipe, and I can put in a name for this variable within my for my data frame by using the variable name on the left side of the equal sign, and then I will put my vector name here. So that will result in new column called as ancestry, which includes the ancestry information from each of the cell lines. So since I created a new data set, probably I want to store it or export it from R. To be able to do this, I can use write CSV function if I want to save this in a CSV format, or I can save it as a text format by using the write table function. So these are the basics for how you can manipulate the data based on your specific question of interest. However, we may need to do additional tasks. In that case, we may need to define some functions by ourselves, and we need to perform this complicated settings, like going through all the data sets. And that's why we need to learn some control structures. So first, look at this another question, that is, we are specifically interested in if the expression of EGFR gene is greater than 5, at least 5% of the cell lines. So to be able to perform this task, first we need to read the gene expression data, and you can see the first 4th rows and 4th columns of this expression data here, as you can see. So first, I need to obtain expression values from EGFR gene. There are alternative ways to do that. First, since I know that it is a first row, I can quickly call the information from first row across all the columns from this object, an expression data object. Or alternatively, if I don't know where is the EGFR gene stored, I can learn it by using the which function. So, and then I try to understand actually which of the cell lines have gene expression value bigger than 5, again by using the which function. That will give me the location of the cell lines within this first row that has a value greater than 5. And to be able to learn how many of cell lines has this information, I can use length function, and that will give me the number of the cell lines that has EGFR gene expression greater than 5. And then I can calculate the percentage in the end, and eventually it is 96. It results in that 96% of cell lines having a gene expression value. So this is important for us because mainly we want to learn coverage, so that we can have enough expression values to perform QTL analysis or any type of differential gene expression analysis, so on. So that is why we probably need to do that across transcriptome, across all other genes. And how we can do that? So we cannot make this one by one, right, for each row. And every time when you write all of this commands, it looks, it's not efficient, first of all, and also it looks very, not regulated, it's not arranged very well. So what you can do is, actually you can define a function which can perform all of the steps within, And you can just provide specific information in that function, and then it will eventually give you the answer of the question you're interested in. For example, in this case, we can define a function by using a function name as function, and X will be your input of your interest. And again, I will replace all of the commands I show you from the previous slide within this our function, and then it will return the percentage of the cell lines that has a certain expression value, greater than certain expression value. In our case, this is 5. So I call this function as get coverage. And to be able to perform this task across all of the genes, I can use a for loop strategy. And for this, I define a for loop that will go over each of the rows of this gene expression matrix. And after I extracted information from each row, for example, in this case, I define it here, and I will provide it as an input for my function get coverage I defined previously. So what is important here is, probably this is something new for us, I put a -1 here so that I can export the first column, which includes information from the gene name, so that I can get only the numeric values. And J indicates the row that will parse one by one through this for loop. For example, it will first get the information from the first row, and then second and third, so on. And I define a gene name as well, so that I can store this information. And to be able to store this information in the end, at the beginning, I define an empty data frame called as coverage values. And every time I extract the information for coverage and also gene name, I store them in a little data frame, and I added this information to the previous one, previous information coming from the previous row by using the rbind, row bind function. Basically, I bind this information from this little data frames by row. And eventually that results in an object called as coverage values that will give me the information from each gene and their corresponding coverage. So after that, after I define this data frame by using, again, some data manipulation tools, I can filter them based on their coverage will be greater than 5. And then I can select only the gene column so that I can learn actually which genes has high coverage. So there's actually even more efficient way doing that instead of using the for loops. And apply family functions can perform this task. For this, we can use apply function, and we provide our data set here as a first item. And second item will be direction of the calculations we're interested in. In our case, we are interested in compute the coverage per row, right, in the direction of this arrow. And we provided this input for the get coverage function each time. So basically, instead of writing a for loop, by providing an apply function, you can let your algorithm parse through all of the rows in your data frame by using a one called line. So in this case, each rows will go through get coverage function, and that will result in the same coverage information. There are other functions called lapply and sapply. So there are differences based on what type of input you can provide and what type of output you can get. In apply function, you can apply function can be applied for either matrices or data frames. One more information here is, so if you want to parse your data frame or matrix through columns, you can replace by 2. And lapply can be used for inputs that are lists and output, again, lists. Supply also can be used for either lists, vectors, matrices, and it will result of this data structure as an output as well. So it depends on what type of a task you want to perform. You can look for other apply family functions, and they are very efficiently parsed through your loop. Now let's go through regression analysis. That is actually the initial question we are interested in. So that was, we want to learn how, what is the variation in the gene expression data, is how much of them is explained by certain variants. So in this case, we can perform regression analysis to understand their relationship between the genotype and the gene expression. In this case, we define gene expression as a dependent variable, basically by using the genotype information, that's our independent variable, we can predict values for Y, which is gene expression value in our case. And to perform this task, we try to fit this data points in a line. And as you see here, we call the regression line. This data points do not perfectly fit the line. So that will introduce an error term. And this line basically selected based on a criteria that minimizes the square sum of each of this error term coming from data set that indicates their distance from the fitted line. So after this line that minimizes the square sum is selected, we see two different variables. One of them is it's for beta coefficients. It included two information, one for beta 0, that is intercept. Basically, that is the point, which is the y value that corresponds to x value, which equals 0. And beta 1, which is actually more important for our cases, that is the slope of that line that indicates how strong relationship between your genotype and your gene expression. So, and regression analysis tests the significance of how this beta value, beta 1 value, is different from 0. And we can perform the linear regression analysis by using R. Let's say I'm interested in to see the relationship between EGFR gene expression values versus this RS 14 and with 76 genetic variants. First, I read my data sets, and then I extracted the corresponding values for EGFR gene expression and genotype value for the SNP. Then I form a data frame which includes information from both gene expression and genotype, and also additionally I added gender and age information. So that resulted in a data frame where you can see the expression values for EGFR and their corresponding genotype value for the specific SNP, and gender and the age of each of the cell lines. So by using LM function, I can perform this task. And basically, my Y now will be in the left side of the tilde. My X will be on the right side of the tilde. And I define those variables by using their column names. And I tell them which data I want to use within this data parameter. And by using a summary function, I can get an information of statistics. So for the beta 1, we can see the estimation values as 0.27 in this second row of the first column. And then you can see the corresponding p-value. Let's say I want to add gender information to this model, meaning that I want to control for the impact of gender on gene expression, and then I want to see the relationship between an expression of genotype. For this, simply I can add gender after plus sign. And as you see here, again, I will look for the corresponding statistics for the genotype. And p-value and beta coefficient are slightly changed now after you add this. So there are some cases your data can be significant after you add for this additional information. We call them co-founders. Basically, the ideal way is checking their impact on gene expression first, and then decide if you want to edit your linear model so that you can perform a robust QTL analysis. Sure. Yes, this is a beta term.
Okay, and then what is the number above that, the 9? What is the...
Oh, this one?
Yeah, what is that?
This is the intercept. This is the beta 0. This is how
okay.
Yeah, basically, generally, we are always interested in the beta coefficient p-value so that we can report them to show the significance of our relationship. Yeah, and then let's visualize our data set finally by using ggplot. So I also want to show how we can look for this linear relationship, excuse me. And ggplot is a very helpful function for data visualization. For this, you use ggplot function, and you write the name of your data, and then you can select the columns which you would like to show within this AES corresponding to aesthetics function. So what I want to do is I want to plot information genotype versus expression values. That's why I put this column names. And I use geom jitter function here so that I can get a scatter plot, which since I have a group of values from 0, 1, 2 for x-axis, but I want to separate these data points. I use kind of a rotation by using a jitter function. Instead, you can use a geom point that will allow you when you have a wide range of x values versus y-axis, that's kind of having an equal number of elements as well. I define a color of this data points here, and then I define size, width, size of those points, and then I name the labels, X and Y labels here. So if I want to add stratify them based on the gender information, what I can do is I can add a color parameter within this AES aesthetics function. It will color those data points based on gender. Or finally, I can add a box plot on top of it by using the geom box plot function. In addition to that, there are a variety of other functions that allows you different visualizations, like for example, bar plot, you can use geom bar. For density plots, you can use geom density, or histograms, you can use geom histogram. Let's go to the final part of our talk today. So as we mentioned at the beginning, Bioconductor is one of the places you can get lots of our packages to be used in, to answer different biological questions. It's a huge storage area that includes above 2,000 different R packages. I just would like to highlight to you a few popular ones. First one is DSeq2 first, that can be used to test differential gene expression analysis. You can use the DSeq2 package named DSeq2 for that. It will perform differential expression by using negative binomial distribution. And you can see this is a famous volcano plot. Excuse me, not volcano plot, but mean expression versus log to fold change plot after you perform this analysis, which is an algorithm generated by Dr. Mike Love and their colleagues. Another package which is I found very helpful is gprofiler 2. Instead of using this web-based tools, you can perform a gene enrichment analysis, gene ontology analysis by using this tool. Basically, after performing your differential expression analysis, you can extract the genes which show upregulation and which show downregulation. And then by using the list of genes as an input for a function called as GHOST within this package, you can find which pathways they were significantly enriched. And it can even perform some visualization for you, summarize what type of enrichment results you have. Finally, this is from my own study with my colleagues. We perform a QTL analysis, and to be able to look at the results, we use some visualization tools. This looks a very complicated graph here. If I can summarize, for example, in this case, we are interested in how an eQTL for coming from certain cell types for the SYNPVW gene is associated with education attainment and global surface area GWAS. So, for example, to be able to make this beautiful and complicated graph, a variety of our packages exist, which includes GWAS, Biomart, and also genomic ranges that will allow you to define your genomic locus with an object, and you can play with them. So yes, thank you so much, all of you, for having me today. And I will be happy if you have additional questions. And if you don't have anything right now, you can also reach me afterwards. This is my email. I'm in the cancer program, living in the 4th floor. Yeah, I will be very happy to talk about my research and R in general. Okay.
Thanks, Nil. That was great. I know for me, I'm used to looking at our tutorials with the built-in data frames like cars. So it was really nice to see it in a genomic context. Do we have any questions in the room? Yeah? Yeah, I could bring you the microphone, or you could come up and stand by one.
Do you have any experience working with data that doesn't fit in memory, and how you would handle that?
Yeah, that's a great point. And for those cases, instead of using my local, generally I don't use my local machine at all for any analysis. We can have a system in Broad called the Uber that allows you to access high memory, computer with the high memory. Basically, you can get an interactive job. You can define how much memory you want, and you can handle those data sets within those additional computers, not your local machine.
Right. Yeah, the eQTL, the last plot, for showing the locus, and how do you know about this snippets, the location in your plot?
Oh, yeah, I'm having the information from how genotype data is stored. That's a software called a plate that give me where the variant is. You can also look at an R as well, because we know there's the genomic location of each of variants. And that you can actually put this information within a G range object in each time for each variant. And basically you create a numeric values of the location of each variant in the genome.
Are these all SNPs correlated with each other, and the encode to the one genes?
Oh, yeah, that's a great point. So in this case, I tested association of all the variants within this genomic locus with SYNPVW gene, only one gene. Exactly.
Thank you.
Yeah, sure.
I'm wondering if you have suggestions for people new to R in these analyses and using ChatGPT or Copilot to try to help them with their coding, or like words of the wise?
Yeah, definitely they are helpful. But as long as you don't know what function does, it can be sometimes dangerous and time consuming. For a starting point, I would definitely suggest going some R tutorials online first. And once you're familiar with, you can also look for ChatGPT, but I think you should be able to judge what ChatGPT does, because sometimes it gives you a mix of functions. Although general, it can be a very helpful starting point as well. But yeah, as long as you have a good R tutorial, sort of a script saved somewhere, you can copy paste it from each time and change it based on your question. So I trust tutorial more than ChatGPT actually.
All right, well, if there's no other questions, let's thank our speaker again.